Hello and welcome to our second video on photosynthesis. I am following the open source textbook out of, I think, Rice University, mainly edited by Clark and Choi and Douglas. And I am doing this because my daughter's taking advanced biology, intro biology at Pepperdine, Sophie. So sorry for calling you out. But this is the second unit in that chapter on photosynthesis. And so you remember that there are light-independent reactions that are part of photosynthesis, like the Calvin cycle, but there's also light-dependent reactions of photosynthesis. So in the chart in the previous video, it's this side of the chart, light-dependent reactions. So it all starts with the sun shines in Pepperdine. So the sun shines. And this is the granny cakes, right? The granum. Each one of these is a woman named thylakoid. Anyway, thylakoid. These are thylakoids. They have chlorophyll in their membranes. The middle of them is called the lumen, the light. It's not really light. It's just basically full of hydrogen ions, protons. But anyway, that's not important right now. So the sun shines. That's where it all starts. For light-dependent reactions, if it's dependent on light, we got to have some light. Now, I've taken this video from, or this is a slide from, I believe, a professor, but it's taken from Pearson Education. And let me remind you that the lovely graphics that I'm using are from the Biology 2E e-text out of Rice, as I said. I believe it's Rice. I need to double check that. But so we have light, and of course light comes in packets, little packages of light. Light is not a continuous wave. Okay, I'm not going there. Anyway, they're little packets of light. If this is a chloroplast, which of course is how photosynthesis takes place in chloroplasts in plants and algae, and there are also cyanobacteria, but that's not important right now. So we have these packets of light. These are the granums and the thylakoid pancakes. And so some light is absorbed into the thylakoid. Some of it goes through, and of course, some of it's reflected. And as we'll say in a second, the color that something has is what's reflected. The color of something is what it can't absorb. So the fact that a tree is, or the fact that a leaf is green is because the leaf can't absorb the color green. It's kind of reversed, that the color something has is what it can't absorb. Well, that's not important right now. So light, like Pepperdine, is a wave. Now, if you walk down, from the beautiful campus of Pepperdine to the Pacific Ocean, you'll see some waves and maybe some surfers. So a wave, the wavelength is basically the length between from crest to shining crest, or you could go from trough to not so shining trough if you wanted. But the wavelength is this, the length between crest and crest, and the higher the frequency, that is the more waves per second, the more energy, right? Because think of it, and I think the book suggests this, that if you're doing jump rope, if you're going slow, that doesn't take as much energy as, and so the higher the frequency, the higher the energy of the wave. So there is this neat spectrum of frequencies. So, for example, radio frequencies tend to be very long frequencies. In the bad old days of AM radio, where we didn't have FM radio, if you drove under a bridge, the radio would cut out because these waves weren't short enough to get under the bridge, so to speak. Thankfully, we don't have that problem anymore. Infrared is basically waves that are in the dark. We use infrared scopes, on all the assignments to get terrorists, that I've been on, which is none. We used infrared scopes, to be able to see at night, to be able to see the longer the longer waves. It's actually a very small part of this electromagnetic spectrum that we can actually see with our eyes, from red being the longer of the light waves, and violet being the shortest of the light waves, Roy G. Biv. But anyway, then of course there's ultraviolet, which we can't see, but it's more higher frequency. X-rays, of course, you can know Superman has X-ray vision. He's able to see through things because he can see the faster waves, and of course the Hulk is made by gamma rays. But anyway, so this is the electromagnetic spectrum, but this little bitty spectrum right here is what our eyes can see, and of course where the range of photosynthesis. So here's the visible spectrum again, from longer frequencies to lighter frequencies, and thank you for the graphic. Okay, so what did Miss Piggy mean? What Miss Piggy meant? Okay, pigment, never mind. Pigments. Pigments are the things in the thylakoid membrane. Pigments are the things that kind of pick up on they convert the photon energy into electron energy. They are the way that the thylakoid captures the energy of the photon from the light. So there are several different kinds of pigments, and each kind of pigment absorbs a different wavelength of light. The two main kinds of pigments are chlorophylls and carotenoids, and they both hide in thylakoid. In the thylakoid is where the membrane actually is where the chlorophylls and carotenoids hide, where they are, in other words. There are two basic kinds of chlorophyll pigments. Actually, there are more than that. There are like 5. But the two most important are chlorophyll A and chlorophyll B, very cleverly named. But actually, I'm glad that they just did that rather than making us memorize a specific name. There are actually 5 children in the chlorophyll family, but A and B are the ones that everybody forgets about the other 3. It's chlorophyll A and chlorophyll B that they get all the press. Carotenoid family has lots of children, and one of the things that the carotenoid pigments can do is, the sun can, on a bright day, there can be a lot of photons hitting a leaf, and so one thing that the carotenoids do is they gobble up some of the excess energy, because if you plug in something to too much power, it'll blow a fuse, it'll blow, things will catch on fire, so what is it in Monsters Inc., remember that, where they're trying to get energy by scaring the child, but they find that if they get the child to laugh, the first time they do it, it not only fills up the thing with energy, but it blows. So the carotenoid pigments, one of the things that they do is they help eat up excess energy when it's just too much. Okay, so what do we see? This is what I was saying a second ago. That what we actually see, the colors we see on a leaf or on a carrot, the carotenoids. What we see is the light that it can't reflect. If it absorbs it, we don't see it. It's when it bounces back that we see it. So a green leaf that bounces back green at us is telling us that I can't handle the green. So chlorophyll A, which is the primary of that color, it can't reflect green, which is why it can't absorb green. I need to fix this. Hold on a second. Just pretend you didn't see that. Chlorophyll A can't absorb green, which is why we see green. Carotenoids can't reflect colors like red, which is why tomatoes are red, or orange, which is why oranges are red or orange, and yellow, which is why corn is yellow. And I have to think that carrots are somewhere in here. Maybe I'm wrong. I'm thinking that carrots have carotenoids in them. I could be wrong. Okay, so in spectrophotometer, so a spectrophotometer is an instrument that can tell you which wavelengths a particular item is absorbing. Spectrophotometer. photometer. And it's taking a picture of the spectrum, right? Okay, so that's what we see. Now it's going to get serious. The rest of the video, it's going to get serious here about this light-dependent reactions. Photons all over the world join hands. An electron train. Join an electron train. Okay, so this is an electron train. Look. Doo-doo-doo. La-da-da-da-da-da. And right. Doo-doo. So the electron train. Okay, so this is a photons come from light packets. Light package. So light packets, photons come into this. These are proteins, right? Don't these look like integral proteins? Integral proteins, remember? Integral proteins are the proteins that span a cell membrane. So here we've got all these little phospholipids, right? That's what this is. It's phospholipid. It's all coming together. And this is I think also a protein called an ATP synthase. But anyway, more on that in a second. But so this is where we're going to have the electron transport chain. And so the photon, which starts with the photon, this is a PS2, which is better than a PS1. No, not really. Anyway, so PS stands for photosystem. Yeah, I knew that. So photosystem 2. The reason this is 1 and this is 2 is because the order of it was discovered. It has no significance. In fact, they could surely come up with a better name. But the PS2 was just discovered second. But that's actually where the process starts. The process starts with the PS2. So first we have a photon, and the photon hits the PS2, and these are the chlorophyll right here, and they get so excited, and they fart an electron to a kind of receptor, and then the electron is kind of passed along on this train through these various other proteins, and the electron gets a little bit tired when it gets to the PS1, and so more photon. So we get another photon, which charges the electron back up, and excites this chlorophyll, and it farts it out again to the receptor, and then we have basically the reduction. Remember, when you add an H to NAD, it becomes reduced. It gains an electron. So as positive ions here, they are oxidized, and so they become reduced, and it becomes a neutral molecule, NAD, which is spit out into the world. Okay, so there you have the light-dependent process. Let's go into more detail. I know you didn't want to, but we have to. So this is photosystem 2. This is where the process starts. So like we said, we have these pigment molecules, like the carotenoids, and we have chlorophyll. This protein is a light harvesting complex, and this is the middle, the lumen, and out here is the stroma. And so what's going to happen is we have a photon, lightning, Thor, and then we might call these antenna proteins, because there any photons out there, and the photon hits it, and then it goes to the reaction center, where the chlorophyll A and I suppose B too, but the chlorophyll A is, and the chlorophyll gets so excited, and it farts an electron out, and the electron goes to the electron receptor. This is called a photoact. Farting is a photoact. Okay, so at the next family gathering, let's be looking for photoacts. So the chlorophyll gets excited. It farts an electron to the electron receptor, and we're in business because now we've got an electron to work with here. And by the way, water is split into its component parts as part of this process. And the hydrogen ions, which is basically a bunch of protons, they end up here in the middle in the lumen. And so you have what's called a concentration gradient, a difference between the concentration outside, which is lower in concentration. So it's going to have it's going to be more basic. It's going to be more higher pH. And then in here, it's going to be more acidic, lower pH, because it has more hydrogen ions. And that's going to create the circumstances to poop out some ATP from this here cell. So that's photosystem 1. Photosystem 1 harvests, it harvests light harvesting complex. It harvests the photon into the pigment here of these are pigments too, into the chlorophyll, which gets very excited and farts an electron to an acceptor, and the electron train begins. Now it's going to be passed along the electron train to photosystem 1, which is called photosystem 1 because it was discovered earlier. Anyway. And so by the time the electron gets here, it's lost some energy because it's passed through some stuff. And so we needed some more photons to get it, to the reaction center, to get more excited. Chlorophyll gets excited and farts that electron on to the electron receptor. And from there, it's going to create the NADP. It's going to reduce, that electron is going to reduce a NADP ion. Okay? So that's photosystem 1. So let's kind of sum everything up here, and then we'll be ready to go almost. So this is where it starts in PS2. This is the electron transport train. And so the hydrogen ions are going to accumulate in the lumen, the photon comes here. The chlorophyll gets excited, farts an electron to the receptor. It's passed along through these proteins, loses some energy, gets to PS1, more photons to the chlorophyll, another electron, and we're going to have, let me move my, and we have NADP ion that becomes a NADP, which is then sent on for the Calvin cycle. Stay tuned next video. Okay, so one more word on what happens because of the concentration gradient. Because we have a low pH with a lot of hydrogen ions inside, and we have a low pH out here, it's going to squirt out hydrogen. It's going to poop that hydrogen right out. And that hydrogen is going to take adenosine diphosphate, ADP. It's going to provide the necessary energy to add another I'm going to wash that phosphate right back onto you. Anyway, and so it's going to be ATP is going to be spit out of this thylakoid cell. Okay, so ATP is pooped out of what's called the ATP synthase. Why is it called a synthase? Because it's it's synthesizing ATP. And this process is called chemiosmosis. Osmosis, you remember, is where something kind of sucks in. This is why you should sleep with your biology textbook on your head, so that it'll soak in while you're sleeping. And so this is a chemiosmosis, which is basically, it's a chemical, it's the chemical concentration difference that's causing the osmosis. At least that's what I'm going with. Well, this has been a long video, our second video on photosynthesis. We'll be back with our third and final photosynthesis video.